Good afternoon for those who've just joined and for those who have been listening to the last three presenters. We're going to continue the subject on emerging therapies, and we're going to we're going to dart around the subject of how service companies are responding to the increased interest in new modalities. Obviously, with the three speakers we've just had, there's quite an ADC. There's quite an ADC focus, so we're going to go back to that in a bit. But the first thing I really want to pick up on is something that Dawn said in her presentation, or something she implied by the data. Monoclonal antibodies. We're talking about emerging therapies, so I get this may or may not be relevant. But they haven't peaked, and they haven't peaked by a long way by the looks of the data. So, I mean, they've been around for 20, 30 years, but can you just kind of talk about how you see, or how we see the industry, how we see monoclonal antibodies emerging or continuing to emerge, so to speak?
I really don't think we have seen the peak. I believe it was Humira sold 18 billion. And if you actually look at some of the small caps like Southwest, eBay, you're selling one product's worth of a company's revenue. I don't think that technology-wise that we're done with monoclonal antibodies. I do agree that I think monoclonal antibodies are still going to be produced and still going to be used as therapies because I don't think our limitations of knowing what we can block or what we can trigger within a disease state is even close to being understood. And I think they're going to continue to be a useful tool going forward.
So, I mean, when we talk about emerging therapies and new modalities, and there's a lot of focus on the cell gene therapies, a lot of focus on mRNAs, microbiomes, ADCs. Really, it's because these are new and unproven, whereas monoclonal antibodies are proven, but there's still a lot of distance to go. So
yes.
Before we do go on to some of those modalities, does Eva, Juan, or Scott have any comment to make about where we stand with monoclonal antibodies at the moment?
Not for me, no.
Actually, during your talk, one thing that I was very interested in is you showed how monoclonal antibodies have continued to grow. One thing I was wondering is if you actually track what the cost to develop and manufacture a conventional mAb, and how that has changed over the past 20 years since we kind of started seeing the introduction of these mAbs. Because I think, obviously, a big hurdle for mAbs is finding the right target, right? Even for ADCs, the antigen is expressed ideally mostly on the target cell, but often it's also on other non-target cells. So I'm just wondering, how the cost has shifted, and if the main hurdle now is just really finding the antigen, or kind of the cost to goods to get into the clinic?
I think that I don't know off the top of my head what it cost then and now. It's actually kind of an interesting point. But I'm pretty sure the cost has come down for that process. And I think it's also evident that there are so many monoclonal antibody companies. There are so many companies that have platforms and develop platforms that it must be a reasonable cost of goods and a reasonable cost to be able to develop and find and screen for all of these targets.
One of the things from a contract manufacturer's perspective that we've seen is customers underestimating the difficulty of getting an antibody that actually has the performance that they're looking for. And sometimes have had to switch manufacturers at the last minute, which has added huge delays in their development. So I think there's almost an assumption on the one side, the chemists assume that the antibodies are easy to make, and the biologists assume that the drug linker is easy to make. So they need to be careful.
Are we? There are so many monoclonal antibodies still in the pipeline, but as far as services firms and CMOs go, is the size of a modality in the pipeline proportionate to the number of service firms servicing that type of modality? So if we look at CMOs, I would guess that the majority are focused in the biologic space with antibodies, because that would make sense. But is that what we're seeing, or is there a kind of a change in the proportions?
I would say off the top of my head, it is a proportional, because if you don't have enough products to be able to fill, your CMO's pipeline, that CMO will essentially cease to exist because they're not going to have enough business. So I do think that it is kind of a proportional case.
Well, it seems to make sense that way. So when we're talking about another modality, antibody-drug conjugates or drug conjugates, I'm wondering, either Scott or Juan, if you can comment on the sort of scope of the market, because there are only 4 ADCs commercially available, but there's 2 ADC CMOs on the panel here, and obviously there are a few others within the industry. But is that proportionate?
There's a company, Hanson Wade, that organizes several meetings, World ADC. They have a software platform that analyzes the industry, and I might be off a little bit on the numbers, but I think there are 160 ADCs currently in clinical development from early phase through phase 3. So there is a huge interest in the area. I think with the approval of Etcetras and Kadcyla a few years ago, the principle was proven, and so a lot of companies have jumped in to investigate that. Small biotech startups as well as big pharma.
I think similar to ADCs is the gene therapy space, with the recent approval of Spark's, adeno-associated virus for application in eye regeneration or regeneration of eye receptors. There is that big shift, I think, towards gene therapy, and companies are trying to grow in that space and just establish at least a presence. I do think that, some of the hurdles that exist in that space of gene therapy lie around the ability to generate a very robust platform process like we currently have for monoclonal antibodies. I mean, from a process perspective, which is what my background is on, we don't have for many of these viral vectors, like lentiviral vectors, herpes simplex virus, we just don't have even the affinity resins that you would need to purify these already complex biotherapies. So I think they're still just in the materials that are available to generate these therapies in a GMP space at, a GMP purity level. There's still definitely a hurdle there as well.
Obviously, gene therapies is such a burgeoning area of the industry that we're going to address that a little bit more later on. I do want to stick with ADCs for the moment, just because we saw the first commercial ADC arrive on the market in 2000 in the form of Mylotarg. I'm curious, between you, Scott, and Juan Carlos, in the almost 20 years since Mylotarg arrived on the market, how has the development of ADCs changed? I'm presuming there's going to be more of an outsourcing focus, but you guys are the experts, so.
Yeah, I'll briefly mention something that Scott and I think both touched in our presentations. I mean, if you look at the conjugation technologies that were used for these early FDA-approved ADCs, they were sort of more straightforward chemistries. Since then, the field has definitely innovated other conjugation technologies, including engineered cysteines that are very site-specific conjugation approaches that generate much more homogeneous therapies. So I think there's definitely been some clear investment and development of very novel conjugation technologies, even shifting towards rather more complex approaches like enzyme-mediated ligations of cytotoxic payloads to very specific locations of the antibody for an ADC product.
I think we're seeing a lot of innovation from the innovators, for lack of a better term, looking at novel antibodies, conjugation methods, or like you mentioned, we've already mentioned site-specific conjugation. I've also seen, there doesn't seem to be a consensus as to what a good drug-antibody ratio is. and it may well depend on what the target and the drug linker, the actual payload is. Some people are looking at very, reducing it down to as 1 or 2 drugs per antibody, and others looking with the, you might recall who's doing it with the glycan conjugation, where they're loading 12 drugs onto an antibody. So that seems to be an interesting scientifically development. And then also in other areas with the antibody fragments, looking at not just whole ADCs, whole antibody ADCs, but also fragments, as seeing if that's a good approach.
So it's fair. Sorry, go ahead.
I was just going to follow up on that point. Yeah, I think we're definitely seeing much different indications, and I think people are not thinking, are no longer thinking of antibodies as only targeting agents, but at least in my experience, I've seen that some indications now take advantage of the increased half-life that antibodies have in vivo, and they use that more as a carrier, non-targeted protein rather than a specific targeting agent. So, for example, if when trying to deliver peptide payloads or even small nucleic acid oligos for like even gene therapies, these antibodies can also serve as very efficient carrier vehicles.
So the antibody bioconjugation technology itself is kind of enabling much of these, some of these emerging therapies. I was writing something about antibody oligonucleotide conjugates the other day, or AOCs, and I've never really heard of those before. And obviously, if you Google AOC, you get quite a lot of hits in a whole different subject right now. But
I think that's one of the, our topic here is how are service companies, dealing with new modalities. I think we're seeing that maybe gene therapy, can also be approached by non-viral vector-mediated vehicles.
Exactly there. So, I mean, your companies, for example, are you easily able to adapt or even just repurpose some of your technologies to feed all these new modalities coming through?
I'll briefly comment on that point. Yeah, I think especially from a conventional up and downstream, recombinant production and purification, when it comes to gene therapies, viral-mediated gene therapies can often look very similar. Although on the upstream side, the gene therapy space and technology is not quite yet using suspension cell cultures that yield high titers. We're mostly limited, at least in my experience, to adherent cultures in somewhat difficult to scale reactors like hyperstacks and cell stacks and the like. But those look very similar to a lot of the upstream production that's used, for example, for vaccines. On the downstream side, viral vector purification is very similar to conventional downstream processing of a recombinant protein like an antibody, where depending on the viral vector being used, you may or may not have an affinity resin, as I briefly alluded to earlier, but you're still taking advantage of chromatographic separations as well as normal and tangential flow filtrations for their purification, which is very similar to adapting those systems or those unit operations from recombinant protein production and purification.
So is this something that Abzena, for example, has actively done?
So I do not believe at Abzena we have actually done development for gene therapies, but I think this is definitely something that could be in the horizon.
And if we go back to you, Scott, talking Carbogen AMSYS, adapting your bioconjugation technologies to feed a whole host of different emerging therapies, is that something that's possible or something that is actively happening?
Yes. In terms of, as I tried to mention, it went by very fast in the talk, but the basic bioconjugation approach itself can be applied to other types of small molecule to large molecule chemistries, such as synthetic vaccines, which is a developing area, where you take a whole protein and link a amino modulator to it to prepare a vaccine, as well as other type of chemistries where you're coupling two different biomolecules with each other for other indications. So there's a lot of, if you have the skill set and the analytical skills there, you can apply it to a whole broad spectrum of these types of large molecules, whereas historically you were only working with small molecules.
If we look at some of the emerging therapies coming through, on the non-conjugation side, the CAR-Ts, gene therapies, the microbiome, Lonza recently partnered and formed a joint venture looking into that specifically. mRNAs, how emerged are they? I mean, is there enough interest in these areas, is enough development that we've seen service companies popping up to support them yet? Or are we really kind of in the early stages of these products?
I don't know.
Dawn will know.
I think we're in the early stages of these products. Definitely, when we think about growth of these sectors, it does kind of feel a little bit like the monoclonal antibody sector back when we first started. Everybody wanted in. There were lots of technology problems we couldn't quite overcome. Being able to produce vast quantities of gene therapies. So I do think that they're definitely, I just lost my train of thought.
Yeah, with
Yep.
Yeah,
Sorry.
I'll just follow up on that and say that also the cost to goods, for example, for a gene therapy is much higher than conventional mAbs. I mean, in my experience, a total process yield for a downstream process that's purifying a gene therapy, if you're at 30% or above, you're super happy compared to, minimum above 60% for a conventional mAb. So some of the quantities that we're producing, at least in my experience, for like phase 1 processes are much smaller quantities than conventional recombinant proteins. But this is also because the indications just require much smaller doses anyway for gene therapies. But I do think there is some ground to be made up in the affordability, right, that some of these new modalities necessitate in order to get to the clinic.
You mentioned earlier Spark Therapeutics as an example. Obviously, when they received FDA approval for their gene therapy, there was great interest within the industry. Does regulatory approval instantly drive investment in services firms to support these things? Is that really proof of a breakthrough, and that will lead to a whole support network looking at those modalities?
My initial thought on that is yes. I think once companies, whether big or small, see that this is definitely space that can grow and has gained FDA approval, I think it definitely incentivizes sort of establishing a presence in that area. But I do think, again, just in my limited experience, a lot of the companies initially, much like Spark Therapeutics, were sort of very small companies that started with the development of these technologies sort of in an academic setting. And now we're starting to see sort of invested from the big players in that area.
So I do want to say, I don't think Spark was necessarily the first one. We had I think it was Glybera, which actually, did prove we could do this, but we just couldn't do this at a fiscally responsible price. And I think that in part, as we work on our technologies and getting our cost of goods down, especially in healthcare today with pharmacy benefit managers, looking at costs, 7 ways to Sunday, I definitely think that once prices have come down, or at least have been modulated by the pharmaceutical companies, as in some cases I've heard them say that they may do this as a mortgage. You're going to be making certain payments to the pharmaceutical company for as long as the therapy works. So I do think that having certain types of structures in place may fuel the ventures, but if we find out that these are all very high priced for very short periods of time, I'm not sure that investment would continue for that.
You mentioned Glybera, but obviously on the sort of cell therapy side of things, Provenge before that suffered from exactly the same cost of goods issues and eventually pricing issues. And I guess the market itself wasn't quite ready to take on these products. It's obviously pricing is going to be a big issue going forward. Avexis, who's about to have its gene therapy approved by the looks of it, they're talking $5-6 million for a patient. I mean, how is industry really going to cope with this change in payment conditions that is obviously going to happen? Has anyone got any ideas here? You just make them. It's anyone in the crowd have any ideas? Because we're talking healthcare providers, we're talking governments, payment issues, and someone's going to have to pay for them. And that seems to me one of the biggest hurdles stopping some of these emerging therapies really taking off. But I mean, perhaps there are some other hurdles that have kind of pulling the brakes on these to somewhat.
Yeah, this is definitely very difficult to say, except when, I hear you put it that way. I think it just incentivizes the development of much more, robust processes. I think that definitely just indicates, right, that at least from a technical perspective, there is so much room for growth, I think, in that area. That's kind of exciting to me.
Do we need to, sorry, go on, Scott.
Just a general comment, I think you see with ADCs, for example, as the example, as the technology has developed, as the clinical success has been demonstrated and now commercial success, I think I hope at least, that you'll begin to see some efficiencies coming in that will make it less expensive. I don't think we're there yet. I think there's still a lot to be developed in that area, and I suspect if you can demonstrate clinically that a medicine is saving lives or extending lives for such a long period of time that there's a return on investment, that then you can justify it. Whenever you have, though, a outside payer paying for the health of somebody else, that's a problem. That's going to be a challenge with any technology.
I suppose almost a paradigm shift on the way people think about paying for these things. If you've got a gene therapy that's a one-off curative disease, then maybe the $5 million, $6 million is more cost effective than a long-term sort of regimen of monoclonal antibody at whatever price.
Yep.
If there are any questions from the audience, now is the time regarding anything to do with emerging therapies and service firms. If not, I have one final question for everybody, and it's looking into the future at the Bioprocess International Theater 2029. What will we be talking about as far as emerging therapies and service firms go? Who wants to tackle that? And there are no wrong answers here.
I think I'll be retired at that time. My company will be very successful.
2029, I think we'll definitely be talking about gene therapy almost as a standard of care for a lot of things. I definitely think by that point we will have done some great things in order to get these therapies to people.
It's tough for me to say what the exact identity of the therapy will be, but I do feel somewhat confident in at least taking I guess that it will be personalized medicine. I think whether it's cell therapy or different therapy, I think personalized medicine is kind of, I think, in the horizon. And in that sense, cell therapy, I think it's a really interesting, demonstration of that.
Well, let's save the date, and I'll see you all then. Thank you very much for joining me. I'd like to thank, obviously, the audience. I'd like to thank the panelists and all the presenters who spoke today. That pretty much wraps it up for the Bioprocess International Theatre for today, but we are back tomorrow with a very full schedule starting at 11. We're looking at biomanufacturing bottlenecks, and we are looking at process optimization in the afternoon, and we have a great set of speakers joining us. So I'd like to say have a great afternoon, and we'll see you all tomorrow. Thank you so much.